Hello everyone. So the next topic which we will be going to study is the auxin. Auxin is basically a plant hormone. So to get in more detail about this, first we have to understand what is plant hormone. According to the standard definition, basically plant hormones are small organic compounds which are synthesized by specific plant cells or tissues that is active in low concentration, and they promote or inhibit the growth and developmental processes of a plant. So this is the standard definition if we go for a plant hormone. Plant hormones are able to exert their action locally or at a distance. For example, some are transported from one organ to another to produce their psychological effects, and some bring about changes in the same tissue or within the same cell when they are synthesized. So basically these are some of the characteristics that have led to the consider that transport is not an essential property of a plant hormone. The site of synthesis and mode of transport for plant hormone is not always clearly localized. So in this figure has shown that all the hormones that are present in a plant. For example, auxin is depicted here, ethylene is depicted here, salicylic acid, cytokines, gibberellins, nutrients. All these things are in a plant, and these are being depicted here. So in the next slide, we will be going to discuss about what are the types of plant hormones. So the concept of plant hormones originated from a classical experiment on phototrophism. So the word phototrophism, which means the bending of plants towards the light, and this experiment was carried out by Charles Darwin and his son Francis in 1880. So he performed this experiment on phototrophism, the bending of plants towards the light. So in this, what he demonstrated? He demonstrated that a signal produced at the shoot apex travel downwards and causes differential cell elongation in the lower parts of the cupitile that resulted in bending towards the light surface. So the plant was bending to the light surface, as we know that the basic requirement to which the plant grows is photosynthesis, that it requires sunlight for its growth and water for its growth. So for this experiment he performed the phototrophism experiment, and he concluded that a signal was produced at the shoot apex. So this signal subsequently shown to be IAA. IAA is what? IAA is indole 3-acetic acid. So it was the first plant hormone which he depicted in his experiment. So a small number of plant hormones have been shown to influence the plant growth and development. So there was a tremendous progress that have been made in the identification of plant hormones, discovery of their effects on plants, and the chemical structures. So based on the functions of or the chemical structures, there are basically these are the 5 categories in which the plant hormones are being classified. First is auxin, which we will be discussing in detail in this slides only, gibberellins, cytokines, abscisic acid, and ethylene. So these are the 5 main groups of the plant hormones. So these plant hormones are basically derived from 3 main types, that is metabolic precursors, that is amino acids, isoprenoid compounds, and lipids. So these are the precursors of the plant hormones. So in the next slide we will be discussing about auxin, that what is auxin, what was the history of the auxin, how it get transported, what are the pathways of the auxin, and what are the various physiological effects and functions of the auxin. So let's get started with the auxin, the history of the auxin first. So as in this figure it has been shown that apical auxin molecules are present at the tip of the plant, and this is the sun which has been showing that here the sunlight is being reached at the tip of the plant, and it leads to the elongation of the plant. As this was the basically experiment of phototrophism, in which the plant was growing towards the light, bending towards the light, it was growing towards the light. So auxin, it was the first plant hormone which was discovered by Fred Swent as a growth promoting chemical in the tip of oat plant, that is oat cupatiles. So this is the scientific name of oat. So the meaning, the auxin meaning, Greek word meaning is to increase basically. And there are various naturally occurring auxins which are known as IAA, that is indole 3 acetic acid, IBA, that is indole 3 butyric acid, and PAA, that is phenyl acetic acid. So this was about the history of the plant auxin, that it was the first plant hormone that was discovered by this scientist, and in the Greek it was named the meaning of in Greek is it is to increase. So this is the structure. As I have already told that there are basically various naturally occurring auxins, and the three shown here, written here, are basically the naturally occurring auxins, and IAA is the naturally occurring auxin in the higher plants. So these are the structures. These are the structures of the two auxins, naturally occurring auxins. And basically the only common feature which is shared by these compounds, that is it is an unsaturated aromatic ring structure, and there is a carboxyl side chain that is being depicted in both these IAA and IBA. This was all about the structure of the auxin. Then what are the functions of auxin? Auxin plays a major role in the plant growth. So these are all the functions of the auxin. It activates the differentiation of vascular tissue in the shoot apex and calyces. It initiates the division of vascular cambium in the spring. It activates basically the cellular elongation by increasing the plasticity of the cell wall. It maintains the apical dominance indirectly by stimulating the production of ethylene, which directly inhibits lateral growth bud. It activates a gene required for making a protein, which is necessary for growth, other genes for the synthesis of wall materials made and secreted by dicytosomes. These basically are all the functions that leads to the that plays a major role in the plant. So basically it promotes growth in many fruits. It initiation and growth of advantageous roots in cutting. There is control of aging in senescence. There's a dormancy of seed that activates the tropic response. Tropic response we will discuss in detail in the next slides. So these are all basically the functions of auxin. Then about the biosynthesis and transport. So major sites for IAA synthesis are the shoot apical meristem, young leaves, developing fruits and seeds. So if we talk about intracellularly IAA, that is found in chloroplast as well as the cytosol. Though IAA occurs in both conjugated and the free form, it has been found that to be conjugated to both high and low molecular weight. So in case of, if we talk about in case of high molecular weight, it is glycoproteins, and in case of low molecular weight, it is glucose. So IAA conjugates are located exclusively in the cytosol. So basically in this we have shown that there are basically two pathways for the synthesis of IAA, that is tryptophan independent pathway and tryptophan dependent pathway. So we will discuss both these pathways in the next slide in detail. Tryptophan dependent pathways. So IAA is structurally related to the amino acid tryptophan. So tryptophan is an amino acid which is structurally very similar to IAA. So it has been shown that IAA is the precursor of auxin biosynthesis. Tryptophan is converted into IAA by several pathways. So this is the pathway, this is the flowchart which is showing that how tryptophan is converted to IAA. Tryptophan first gets converted to indole 3 pyruvic acid, then it gets converted into indole 3 acetaldehyde, then it converts into indole 3 acetic acid. So basically tryptophan is the precursor of auxin biosynthesis, and this is how the whole process of conversion of tryptophan to IAA through this pathway involves deamination of tryptophan, tryptophan, decarboxylation of 3-pyruvic acid, and oxidation of 3-indole 3-acetyl. These are all the things which have been discussed here. So tryptophan leads to the uh, leads to the indole 3-acetic acid formation. Then next we will talk about the tryptophan independent pathway. So there were many recent genetic studies that have provided the evidence that plants can synthesize AAA, that is indole 3-acetic acid, via one or more tryptophan independent pathways. So this route does not involve tryptophan directly, but as a precursor in the formation of auxin. Okay, then a tryptophan independent pathway of IA synthesis starts with indole that has been partially characterized in various plant species. The indole has been characterized in various plant species. However, the precise pathway for tryptophan independent IAA synthesis is not known. Not much detailed study has been done on IAA synthesis in tryptophan independent pathway. So this is a very short topic which is to be covered. Then there are many uh, physiological effect of auxin, that is cell elongation, cell differentiation, rooting, apical dominance, fruit set, and tropic response. So we will discuss each of these in detail in the next slides. So cell elongation. In case of auxins, they stimulate cell elongation or expansion in stems and colipetals. So basically, it's, what is the function of auxins? So auxin stimulates the cell elongation and expansion in cell and cupetals. They promote the elongation growth by increasing the cell wall extentability. Okay. So auxin stimulates proton pumping by activating or inactivating proton pumped H plus ATPase present on the plasma membrane of this plant. So this is how the role of cell elongation is played uh, in case of plants in case of plants. Then cell differentiation. The differentiation is basically of relative amounts of xylem and phloem that are regulated by auxin concentration. For example, if there is a high concentration of auxin, it leads to the differentiation of xylem, and if there is a low concentration of auxin, then it leads to the differentiation of phloem differentiation. Okay, then next will be rooting. So rooting, what does auxin? How does auxin play a role in rooting? Auxin basically inhibits elongation of the primary root, but stimulates root initiation on stem cutting, that is advantageous roots, and lateral root development. So the lateral roots, basically, what is the lateral roots? Lateral roots are basically commonly found above the root hair zone and originates from cell present in the pericycle. So auxins stimulate these pericycle cells to divide, and the dividing cells gradually give rise to the lateral root. So this is how the process goes in the process rooting. Then apical dominance. What is apical dominance? First, we have to understand the definition of apical dominance. The growing of apical buds inhibits the growth of the lateral buds. So this phenomenon is called as apical dominance. Removal of the shoot apex usually results in the growth of the lateral buds. So plants with strong apical dominance are typically weak branched and show a strong branching response to decapitation, which is decapitation means removal of the shoot tip. So if we talk about the apical dominance, then in the presence of apical bud, the outgrowth of the lateral bud is inhibited by auxin. So auxin maintains basically the apical dominance by stimulating the strigogalacton synthesis pathway. So it leads to maintain the apical dominance by stimulating this synthesis. Then the next is about the fruit set. What is fruit set? Auxin promotes the fruit development. Fruit set is basically defined as the transition of a ovary to a rapidly growing young fruit. So auxin promotes the fruit development. After fertilization, fruit growth may depend on auxin produced in developing seeds. Auxin is produced in the endosperm and the embryo of the developing seeds. In some plant species, seedless fruits may be induced by the treatment of unpollinated flowers with auxin. So auxin play a major role, and the production basically auxin leads to the production of seedless fruits. So auxin plays a major role in this also. Then tropic response. Auxin induces differentiation growth in plant hormones under the influence of directional stimuli, that is light, gravity. This is called the tropic response. So uh, if someone asks you what is tropic response, so tropic response is basically when the plant organ under the influence of directional stimuli, that is light or gravity, then it is called as tropic response. So these are some of the references from which the slides have been made. I have cited the papers. Thank you.